everyone. Thank you for joining us for a presentation titled The Varied Application of Dried Blood Spot DBS Sample Collection During the COVID-19 Pandemic. It's my pleasure to introduce you to Dr. Zahidul Mondle, Lead Technical Product Specialist, Cytiva. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Now, if any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following the end of the presentation. Please join me in welcoming Dr. Zahidul Mondle. I will now turn the presentation over to him.
Thank you for a nice introduction. The title of my presentation is The Varied Application of Dried Blood Spot During COVID-19 Pandemic. Our mission is to advance and accelerate therapeutics. My presentation will cover how sample collection device advance and accelerate faster diagnosis, the first step of therapeutics. Here is my agenda for this talk. I'll talk about what is sample collection card with a touch of historical aspect of sample collection and diagnosis, applications of 903 card, where I will discuss how this sample collection card reduce risk, monitor surveillance in cost-effective manner. I'll bring few examples and standard protocols of Cytiva, sera extractor, nucleic acid microextraction from sample collection card. Finally, I will discuss your considerations where to apply this technology. Let's start with sample collection card. There are many names of sample collection cards. Whatman 903 card, Whatman sample collection card, dried blood spot card, or DBS card are alternatively used that speak to the same purpose. Occasionally, it is referred to as Guthrie card. The 903 cards rose to prominence in 1962 after Dr. Robert Guthrie first published procedures for screening of newborn babies for phenylketonuria. Phenylketonuria, a disease still affects 1 in 12,000 people, where phenylalanine dehydrogenase that converts phenylalanine to tyrosine is defective. As a result, phenylalanine accumulates on the surface of blood-brain barrier and impair other nutrient transport to the brain, impairing brain's ability to protein synthesis. Since then, Guthrie card samples from 2 to 9-day-old babies have been collected routinely, not only for phenylketonuria, but also for congenital hypothyroidism and sickle cell disorders. The detection of markers of other diseases, such as human chorionic gonadotropin in Down syndrome, glycated hemoglobin in insulin-dependent diabetes, and the estimation of drug levels, abuse of drugs, have also been investigated. The limitations of sensitivity and specificity when screening such small volume of blood, such as 5 to 10 microliter, restricted the use of dried blood spot for many years. However, recent advances, such as the production of monoclonal antibodies, expression of synthetic proteins, and the introduction of the polymerase chain reaction, or PCR, have overcome many of these problems, allowing the potential of a vast bank of stored DBS to be useful by biochemists, geneticists, and microbiologists. Now, the DBS system is the standard for testing antibodies or proteins, viral RNA or DNA, bacterial RNA or DNA, or human animal RNA or DNA. The World Health Organization recommended procedure for viral load results obtained from plasma and DBS have proven to be comparable. Recently, FDA approved the Vibrant Antibody Chemiluminescence assay for differentiating SARS coronavirus to IgM and IgG antibodies on Whatman 903 card. What was the Guthrie card at that time? Guthrie card was basically S&S or Slaker and Schwell 903 paper, and hence it took the name 903 card. DBS cards are made from 100% cotton linter with no additives. Workman carefully ensures homogeneous composition, uniform thickness, and absorbency, which ensure consistent sample recovery and test performance. Capabilities of Workman 903 cards. 903 card is a stable and cost-effective method to obtain blood samples on 5-spot protein saver cards as shown on the left 2 pictures. 5-spot perforated protein saver cards in the top middle, protein saver snap apart card in the right. Moreover, we offer any custom size cards for diverse volume of blood. Dried blood spot card can be provided with scannable barcodes, maybe numbering system, or with color code. The blood spot should then be allowed to air dry and ideally stored in low gas permeable bags that contain desiccant to reduce humidity. Samples can be tested immediately, stored at ambient temperature, even under tropical conditions, or frozen at -20 degrees Celsius, where the level of IgG antibodies may remain stable for several years. Some accessories necessary for the dried blood spot is drying and packaging. Drying rack is convenient tools. Workman drying rack for 903 protein saver card is easily assembled, can be affixed to a wall or countertop with optional hook and loop stickers. Foil pack and desiccants for easy shipping for remote analysis. So now diverse application of DBS. So intended uses for DBS samples. DBS obtained from children and adults have been used for the diagnosis of infections and for sentinel surveys in many parts of the world. Surveillance. Surveillance detect acute or past exposure or infection. For surveillance, both molecular and antibody testing can be used. Surveillance was World Health Organization's mandate for HIV infection surveys. Now, in this pandemic, World Health Organization is trying to understand from COVID-19 pandemic surveillance how much of the population has been infected with the SARS coronavirus 2 causing COVID-19. How is this changing over time? Are there different characteristics or risk factors that are associated with SARS coronavirus 2 infections, such as age, location, or underlying health conditions? How many residents experience mild or symptomatic COVID-19 illness? How long can antibody be found after COVID-19 infection? Also, case management of suspects, detect active infection. For case management of suspects, molecular or antigen testing can be used. Contact tracing, detect asymptomatic or symptomatic acute infection. For contact tracing, molecular and antigen testing can be used. Monitoring response or recovery. For monitoring response or recovery, both molecular and antibody testing can be used. Prognosis, maybe cytokine response sometimes gives the clear answer, but for environmental monitoring, only molecular detection is needed. There is World Health Organization mandate about sample types that allows plasma, serum, whole blood, fresh or frozen, or DBS sample. In any sample types, the sensitivity and specificity must meet regulatory guidelines. So other applications of DBS has been proven to be useful for hepatitis C, hepatitis B, drug resistance, viral load monitoring, human immunodeficiency, sexual transmitted disease, bovine anti-Brucella abortus detection, malaria, tuberculosis. For example, about surveillance of HIV, actually, in the beginning of this surveillance, it initiated a huge doubt over the sensitivity and specificity of DBS-based tests. Compared to plasma or serum, DBS contains host cells, protein, and nucleic acid, whereas plasma or serum has a kind of pure nucleic acid from infectious organisms. So the DBS was not out of doubt about its sensitivity and specificity. The other point was the volume of sample from DBS is limited to 5 to 10 microliters, so the chance of false negative result was another doubt. But DBS is compared with serological assay by parallel testing of DBS and sera, which showed 99 to 100% concordance. Furthermore, laboratories at the Centers for Disease Control Atlanta extended the screening quality assurance program to include test panels of anti-HIV positive and negative DBS, which were distributed throughout the USA and overseas. Subsequently, a similar quality control program was made available by the Central Public Health Laboratory in London. Since the inception of anonymous screening, several millions DBS have been tested, enabling accurate data to be obtained about the prevalence of HIV-1. Similar surveillance has been done for human T-cell leukemia or lymphoma virus. It's called HTLV. HTLV-1 has been recognized as the causal agent of adult T-cell leukemia. Transmission may occur by several means, including breastfeeding and through contaminated blood. In order to assess the extent of HTLV or human T-cell leukemia lymphoma virus, in women of childbearing age, a modified gelatin particle assay costing very little per test was used to screen over thousands DBS obtained from neonates. Another example of surveillance was done for Toxoplasma gondii. Toxoplasma gondii is recognized to be a major cause of congenital infection, which may result in mental retardation. However, the prevalence of infection varies widely in European countries. A modified latex test consisting very little per test has been used for extensive surveys to determine the maternal prevalence of antibodies. I should talk about detection of nucleic acid in DBS. The potential for the use of DBS for the detection of nucleic acid was first realized in 1987. When DNA was released from the filter paper by microextraction, the molecular analysis of DNA obtained from neonates has enabled detection of genetic mutations responsible for cystic fibrosis, oncogenesis, markers of type 1 diabetes, and thalassemia. The stability of DNA has made it possible to apply PCR to detect proviral HIV DNA sequences several months after sample collection, even when DBS had been held at room temperature. Recent studies have shown that HIV DNA and RNA can be recovered after 5 years without refrigeration. An unexpected benefit of storage is that inhibitors of PCR, such as protein, hemoglobin, and iron, seem to become increasingly resistant to elution into the sample, whereas nucleic acid seems to aid stability and does not impair its elution. So viral load monitoring for HIV, this is a practical example where it is being used. So Ministry of Health, Global Health Organization, a part of CDC, and National Institute of Health dried blood spot samples have been used for the detection of severe acute respiratory syndrome coronavirus 2 spike antibodies. DBS sampling offers an alternative for population-wide serologic testing in the coronavirus pandemic. For viral load monitoring, key products used are 903 cards or DBS cards, accessories such as drying racks. So another example of use of 903 card in National Scientific Institute Medical Research in Wales, where scientists have developed a convenient low-cost antibody test for COVID-19 based on dried blood spot sampling technique. The novel antibody test uses a single drop of blood and blot it on a specialized 903 cards. The cards can then be shipped to laboratories for testing. Once they reach the laboratory, a disc is punched out from the card and the antibodies are released using a specialized liquid called extraction buffer. They have optimized the enzyme-linked immunosorbent assay, or ELISA, antibody test to be used with the DBS method, and translating the test onto an automated platform will allow hundreds of samples to be tested at once. Now, I'll give you some examples of collection protocol. Sample collection by a skin puncture. Blood can be collected from skin puncture or venipuncture. Venous blood is the specimen of choice for more routine lab tests or reference ranges, but skin puncture blood is preferred for remote sample collection or when small amount of blood is adequate for the test. And the specimen of choice for pediatrics, elderly, severely burned patients with thrombotic tendencies, extremely obese, apprehensive patients, and home-based test. Small amount of blood is easier to manage in remote areas such as home or clinics. The collection on DBS is pretty simple. Right after finger prick, apply pressure, allow a large drop of free-flowing blood to collect, quickly and gently touch card with blood. Let blood soak through. Repeat until at least all circles are filled. Be sure fill out the name and date or other special requirements on the cards. Once the card is dried, they are ready to ship. Now, what are the advantages of DBS? Number one advantages is reduction of risk. The collection of capillary blood or filter paper is simple to perform, requires minimal training, and doesn't involve the risk associated with use and disposal of needles and syringes. The DBS represents a low infectious hazard, as some viruses such as HIV 1 and 2, human T-cell leukemia 1 and 2, and hepatitis C virus that are known to be present in serum or plasma, but they lose infectivity due to disruption of their envelope on drying process. However, virus in high titer may remain viable on a surface for several days, and hepatitis B virus may remain infectious for at least 7 days. Risks associated with shipping are minimized, as DBS cannot leak or be broken in transit, and there is no requirement for carriers on dry ice. Handling of potentially infected material is also reduced, as there is no need to centrifuge and separate syrup from blood clots. There is another economic advantage. Collection and processing of DBS is considerably cheaper, at least 3 to 5 times cheaper, than samples collected by venipuncture. DBS can be readily shipped in sealed envelopes to reference centers, whereas sera need to be packed in break-proof containers. Transport of frozen sera may require the use of dry ice or liquid nitrogen, requiring further specialized handling and adding considerable weight to the item. The majority of the diagnostic technique, for example, particle agglutination test and enzyme immunoassays, which have been used to screen for antibodies in DBS, are simple and generally require little capital expenditure. The use of the agglutination test is particularly cost-effective as they can be performed in the absence of an electricity supply. This is ideally suited to countries with limited health budgets. And surveillance for large-scale epidemiological surveys can be performed rapidly, enabling strategic decisions to be made about targeting at-risk population. This may be particularly valuable for monitoring herd immunity and the extent of vaccine coverage at a time of eradication. Cytiva Genomics and Molecular Solutions optimize DBS card sample collection and elution for molecular diagnostic of SARS coronavirus 2. The process is pretty simple, using only application of 20 microliter of blood on 903 cards, dry for about 3 hours in room temp, remove 12 millimeter punch from 903 card, homogenize and extract sample using the sera extractor virus pathogen kit. So what is sera extractor virus pathogen kit? This kit has been developed in response to global COVID-19 pandemic and aimed to address current challenges faced with supplying kits for testing and scientific research. The format of the kit and a rapid protocol provide an excellent high-throughput solution that should aid in global efforts of scientific and medical community to detect and study the virus. Sera extractor virus kit offers the advantages of solid phase extraction using magnetic beads. Nucleic acid samples are allowed to bind on magnetic bead surface. This is called solid phase surface, washing out the unbound samples and eluting pure nucleic acids ready to run the test. The protocol can be adapted for both manual and automated high-throughput processing. Rapid extraction procedure can be done in less than 30 minutes using diverse sample matrices such as blood and universal transport media at low concentration. Sera extractor virus kit is compatible with molecular biology techniques including quantitative polymerase chain reaction, droplet digital PCR, and next generation sequencing. In addition, a recent study found that on cardboard, no viable SARS coronavirus 2 was measured after 24 hours. So this is the result for the detection of SARS coronavirus 2 by sera extractor virus pathogen kit from 903 card. In this graph, SARS coronavirus 2 is diluted in coupon universal transport medium with human cells. RNA extraction done from 903 card within 24 hours, which we can call T0 in the left 3 extraction of the graph. RNA extraction from 903 card within 14 days, we can call it T14, shown last 2 extraction of the graph. So T0 and T14, so the first 3 graph and first 3 bars and last 2 bars were compared with liquid samples in the middle 2 extraction. PCR amplification was done using N-on nuclear protein primer sets and RNase negative control. As per CDC guidelines, only samples for which the N-on marker crossed the threshold at CT values were considered positive. So no amplification in the N1 COVID-19 specific primer set were observed in unspiked controls. Unspiked negative controls were not shown in this graph. In this slide, whole human blood stored at room temperature on Whatman 903 cards for 24 hours, which is called T0, and 14 days is the T14. CT values obtained for the N1 COVID-19 specific primer set with 50 copies per microliter or 1,000 copies per extraction of the heat inactivated viral RNA in the input sample and human-specific RNA-sp primer set are presented. Error bars represent standard deviation. Note that COVID-19 specific amplification was not detected in any of the unspiked control samples. CT undetermined, data not showed. So the sero-extractor virus kit provides efficient total nucleic acid recovery from different samples type and in different concentrations with high sensitivity in downstream detection. So here are some important considerations before using Whatman 903 card. All Whatman 903 card, protein saver cards, are marked for research use only, meaning you need regulatory such as FDA, UA, or country-specific approval to incorporate a DBS card into your test. Let me answer some common questions. How does 903 card work? Actually, it's for biomolecules that are bound to the cellulose fiber matrix, capturing the sample for a one-word analysis upon extraction. What sample type can I apply to 903 card paper? Typically blood, but any bodily fluid can be applied. How should I store the unused 903 card? Recommendation for the storage of 903 cards is at less than 25 degrees Celsius and 60 degree relative humidity, protected from direct sunlight. What is the shelf life of an unused 903 card? Is 5 years, very long shelf life if it is in Cytiva package. What puncher can be used with 903 card? Any puncher, mostly used, any puncher can be used. What accessories such as puncher and mats does Cytiva provide? We do not have any punches we offer, but do have pouches and desiccant we can offer to make up a kit. What happens if 903 cards get wet, either before or after storage? If the 903 card gets wet prior to sample collection, discard the unused wet card. If the product gets wet after sample application, take a punch from the specimen as soon as possible and continue processing. How do I apply the blood or other liquid on samples onto 903 cards? Products are provided with an protocol showing how sample should be applied. Can I reapply sample to the card after the spot has dried to get more sample onto the 903 card? Not necessary, because the amount of sample you will have for first application, you will be good enough. Can custom formats be made? Yes, our design team can work with you to produce a device that will fit your needs. Right. Can you apply previously frozen blood to 903 cards to purify sample? Yes, a larger punch may be needed, though, as some of these cells may have been lysed on freezing. How many punches can I get from one 903 sample circles? Depends on the punch diameter and circle size. You can determine. Can I elute DNA from 903 card? In principle, yes, with the help of proteinase K treatment and a phenol chloroform isoamyl alcohol at a 25-24:1 ratio extraction. Yes, this application is quite possible. What do you mean by excessive humidity during storage? What about humidity during shipment of spotted and or unspotted product? Humidity may compromise the sample. Biomolecules degradation could occur. There, we recommend to use a sealed bag containing a desiccant packet. How much blood does each card hold? That's a good one. This depends on number of circles per card. Each circle sample area holds 75 to 80 microliter of sample, maximum 400 microliter per cards with 5 sample areas. What certification is available? Certificate of analysis is available for 903 paper used for each job. We normally do not provide job-specific certificate of analysis. Contrary of origin certificates is supplied for export countries as a mandatory document. Are MSDS available? No. Material data safety sheet are available as no chemicals are not available, as no chemicals are used for this product. What types of barcode can you print? Cytiva can print most barcodes that are utilized in the industry using high-speed laser printers. Cytiva can customize to any barcode size and is also able to print 2D barcodes. Can you print barcodes on 903 filter paper? Because of the nature of 903 filter paper and its high absorbency, Cytiva does not recommend trying to print barcodes directly onto the 903 paper. What is your typical delivery? The complexity of the card and volume can affect delivery. However, the majority of orders can be processed and shipped 6 to 8 weeks after PO receipt and customer copy approval. That was my last slide. Let's keep in touch. Thank you very much for your time. I can take your questions.
Dr. Mondle, can you hear me okay?
Yes.
Very good. Okay, sounds good. Okay, well, Dr. Mondle, I want to thank you for that outstanding presentation. We will now move into the live Q&A portion of our webinar. Now, as a reminder to our audience, please submit your questions via the Q&A box. Okay, we have lots of questions, so let's just jump right in. Our first question: how can I get more detail on the protocols of how to use 903 cards in the many applications you've mentioned in this presentation?
All right. Let me get the question from the bottom.
Sure thing.
All right. Just a second.
Sure.
All right. I get protocols on how to use 903 cards in many applications you have missed in this presentation. You have many applications, for example, nucleic acid test or antibody test or some specific biomarkers, depending on the type of samples you're collecting. Say if it is blood samples, so there are 3 ways you can collect the blood: finger pricking, or venipuncture, or arterial puncture. And you also can collect any kind of biofluid like urine, saliva. So depending on your sample type, your collection protocol will be different, but the process is very simple. We have the protocol that how much sample can be applied per circle. Depending on that volume, you can use the certain volume to apply on the 903 card. After drying for 3 hours at room temperature and you can collect the punch using a puncher and elute your samples. And sample elution or sample preparation is variable depending on the type of test and compatibility of your reagent. But the straightforward process is just punch the cut out, and elute your samples into a right buffer, which is suitable for your test.
Excellent, thank you. Our next question, can you speak to other sample collection methods that you've used?
All right, I already discussed the finger pricking sample collection. Involves taking a very small amount of blood from the patient, usually from the end of the finger. But arterial sampling, which is close to the palm, and this blood sample commonly takes place within a hospital environment, arterial sampling. It is used in the identification of metabolic, respiratory, and mixed acid-based disorders where carbon dioxide levels require understanding or monitoring. There is an increased risk of bleeding complications in patients with coagulopathy. But venipuncture is most common way to collect blood from adult patients from a superficial vein in the upper limb. This vein is close to the skin and doesn't have many large nerves positioned close by. This reduces pain and discomfort for the patient. There are also risks related to the storage, transportation, and potential loss of contamination of the blood samples once they are collected. So other than blood, saliva, urine, or any biofluid can be collected on sample collection card.
Thank you. Let's go with this one. Are there use case examples in this pandemic that you can speak of?
Oh, definitely. Recently, FDA approved the Vibrant antibody chemiluminescence assay for differentiating SARS coronavirus to IgM and IgG antibodies on Whatman 903 card. World Health Organization is trying to understand from COVID-19 pandemic surveillance. World Health Organization has approved DBS system for HIV surveillance and now stepping up with DBS sample to understand SARS coronavirus 2 as well. And also, world scientists have developed a convenient low-cost antibody test for COVID-19 based on dried blood spot sampling technique. It's also an automated ELISA format. So these are the common examples, but we have been working with many customers who are trying to DBS card to incorporate into their lateral flow system as a sample collection device and lateral flow test for the detection of that sample. And finally, probably this lateral flow with the DBS card will be approved by FDA or other EU system. Yes.
Excellent. Thank you. Now, how long can samples be stored, and what are the best storage conditions? Once
after sample collection, depending on the analyte target, it could be antibody, nucleic acid, the length of sample stability may vary. For example, nucleic acid, DNA, and RNA. RNA is unstable on any sample, on any condition, in any condition, either liquid or in dry blood spot. So samples can be tested immediately or usually can be stored in ambient temperature, even under tropical conditions, or frozen at -20, -80 degrees Celsius, where the level of IgG antibodies may remain stable for several years, but the stability of DNA and RNA may be variable but it has been tested by PCR and DNA sequences several months after sample collection, even when DBS had been held at room temperature. Recent studies have shown that HIV DNA and RNA can be recovered after 5 years without refrigeration as well.
Excellent. Dr. Mondle, as swabs are in short supply, could 903 cards be used to collect saliva, mucus swabs for COVID-19 antigen detection?
So it has not been tested, but saliva antigen for COVID-19, definitely it can be collected on a 903 card, and the customer has to evaluate. This is kind of new antigen. We don't know many characteristics. Customer has to be evaluate the stability of the 903 card with SARS coronavirus 2 antigen.
Thank you. And we have time for a few more questions. Let's go with this one. Are there any higher contamination risks as compared to other sampling methods?
Definitely, finger pricking is much safer than any other methods of sample collection. As we know, phlebotomy, the drawing of blood, has been practiced for centuries and still one of the most common invasive procedures in healthcare. Each step in the process of phlebotomy, such as collecting blood, shipping, opening the tube, and take the samples for testing, affects the quality of the specimen and involvement of human hands work, which caused the error in many ways. And it also caused the injury for the patient. For example, the touch of a finger to verify the location of vein before insertion of the needle increases the chance that a specimen will be contaminated from the hands. And these can cause false results, prolong hospitalization, delay diagnosis, and cause unnecessary use of antibiotics. Also, jostling and jarring of the test tubes in the transit can lyse or break open red blood cells, causing false laboratory tests as well. So similar finger pricking and 903 card sample collection, even though it is safer, but definitely it requires human involvement to clean the fingertips and drop the blood. So the precaution should be taken for finger pricking as well, but this is definitely much safer than venipuncture blood collection.
So piggybacking off on that question and answer, can you elaborate what the common root causes are when sampling is done incorrectly using these cards?
Right. The root causes could lie in every steps that human or the sample collecting person is taking. People not storing the product correctly, even after sample collection, and is not transporting in timely way to the lab. Say finger pricking, at the time of finger pricking, finger must be clean. Apply pressure during, say, blood is not dropping out and you need to apply a tiny pressure. During pressure, if your hand move away from the circle, then you might spot somewhere else, and allow a large drop for free-flowing blood to collect. Sometimes when the circle is full, you have to remove your hand away. So during the touching, when the blood is dropping from the finger, the card should be in contact with blood, and is a kind of tricky, and you have to maintain this properly. And and also the drying condition. If the blood is not completely dry, and if you put in a package, so the blood might contaminate some other area of the card, even the envelope, which could be risky for the handler. So the drying condition is one of the critical. And then you have to fill out the name and date of other date and some other special requirement. Once the card is dried, it is ready to ship, and then you have to ship it properly. So there are many steps involved. So every step could be faulty if it is not accurately maintained.
Thank you. That's some great information. We have time for one more question. Let's go with this one. I need to track and automate my sampling process. What options do you have?
All right, dried blood spot card can be provided with scannable barcodes, which we are able to customize upon request. We are also working in development to help create a framed version of the 903 card, which is compatible with automation equipment such as the Hamilton EasyPunch Starlet. More information is coming. Please follow once available.
Great. Thank you so much. We are out of time today. But I want to tell our audience, don't worry, because those questions that were submitted and we were unable to get to, they will be answered via email. And we have your registration information in your email. Dr. Mondle, thank you again so much. And thank you again to our audience for their outstanding questions.
Oh, thank you so much. For your cooperation, and you have been working so hard from the beginning of this conference and for this presentation. And thank you, my audience, for listening. If you have any question, please feel free to contact me directly. We'll be happy to answer your questions.
Thank you, Dr. Mondle. Now, I want to tell our audience as well that today's presentation is available for on-demand viewing for 12 months. So please remember to share this with your colleagues who may be interested in this topic. You may also submit questions during that on-demand period. Now, don't miss out on the other presentations on our agenda. Those that have already presented live are now available on demand. So visit that agenda tab in the auditorium for a full listing. Thank you again for your participation. And Dr. Mondle, thank you again for your time today.
Thank you very much.
Have a great day.